Hello, and welcome to the final installments of Labrats. So last week we did an experiment where, well, it was kind of in two parts. There was a biological aspect to the experiment, and then there was a chemical aspect to the experiment. So initially what I had you do is to take a chicken egg and place it in vinegar and dissolve the shell. So the reason that vinegar is able to dissolve the shell is because vinegar is an acid and it reacts with the calcium carbonate in the shell. And I didn't tell you this last week, but that's actually the reason that there's a bunch of concerns about acidification in the oceans. Like you probably have heard about the coral reefs being in trouble, and that's because essentially the oceans are becoming more and more acidic, and as you become more acidic, it dissolves calcium carbonate, and that's actually the same reason why coral reefs are kind of having problems right now. So it's that same principle that allows the calcium carbonate in the eggshell to dissolve, is that basically the vinegar, the acid in the vinegar reacted with the calcium carbonate. It released carbon dioxide, which were the little bubbles that you would have seen on the egg as it dissolved. And if you took that egg out and wiped it, you would notice kind of a filmy residue on it. That was actually the calcium that was reacting out of the calcium carbonate. So eventually you would have ended up with a egg that did not have any shell at all, and you would have put one of those eggs in a very super saturated salt solution, and you would have put one of them in water. And after a few days, you would have taken them out and noticed that the one in water was much smaller than the one that you put, or pardon me, much larger than the one that you would have put in salt water, which would have been much smaller. And the reason for that is osmosis. So osmosis is essentially the movement of water across a semi-permeable membrane. So this is kind of like the cells in our body, where it will basically passively move water in or out of a cell depending on whether there is more solutes on the outside or the inside. So essentially how osmosis works is water will preferentially move towards an area of more solute concentration. So if there's more salt on the outside of a membrane, the water will move out and into the outside area. So that's essentially how this works, is that water wants to move towards, a more salty environment is essentially how you can think about it. But it goes far beyond salts as well. It's basically any dissolved solute, so anything that's dissolved in the water itself. And this actually plays into what we're doing this week, which is our final experiment, which is actually going to be extracting our own DNA. So DNA is basically short for deoxyribonucleic acid. And what that is essentially DNA codes for every single trait that we have. Not even just the traits that we physically express, which is called our phenotype, which is how we look on the outside, but also our genotype, which are all the genes that we carry and can pass along to our offspring. So essentially, DNA we've discovered is unique to every single organism on the face of the planet. So, not only is DNA unique to humans, it's unique to every single person who's born, because you get half your DNA from one parent and half from another, so it's unique to you. And so you can use it for all kinds of things. It's being used for criminal investigations, for understanding how certain diseases that are passed on from one parent to their offspring work. It's used for all kinds of things. So this technique that we're going to do is actually truly how DNA is sourced to do these kinds of experiments. Even discovering how certain organisms are related to each other is all based on DNA. And so what you need is the first thing that you need is a sports drink. So in this case, product placement Gatorade. And the reason we use sports drink is actually related to the whole osmosis thing. So you need an isotonic solution. And what isotonic means is that this solution is at the same concentration as our cells. So water is not going to rush into or rush out of our cells when I put this in my mouth. It's going to be balanced with the solutes that I already have in my body. So it'll keep the cells intact, essentially. There will be no osmosis that takes place. And that is really important for this experiment. So the first thing you do is you take sports drink and you actually put it in your mouth, and you don't swallow it, you just swish it around. So you basically chew the sides of your cheeks down, and you put a bunch of your saliva is going to rush into it, and you're going to end up with a bunch of your genetic information in this cup. So the next thing that you're going to do is you're going to take the test tube that I provided to you, and you're going to put about half of this solution in this test tube. And the next thing that you're going to do, so that's about half of that solution. We'll just set it to the side. So in your saliva will have your DNA. Also, the reason why you kind of chew your cheeks a little bit, not so much that it hurts you, but just to kind of scrape the sides, is because you have a lot of cheek cells. A random piece of information is that the cells in your mouth actually replicate about 4 times faster than anywhere else in your body, because you're constantly basically like there's a lot of abrasion and stuff that takes place because you're eating food all the time. So your cells need to replicate 4 times faster in your mouth than anywhere else in your body. So that's just an interesting fact. That's why also like sores in your mouth end up healing way faster than like a cut on your arm, is because your cells actually are able to heal 4 times faster in your mouth. So interesting fact. So you're just gonna take some dish detergent and you're gonna put it inside of this, and then you're gonna close the cap. So remember how we learned that when we did our lab last week, we learned that cells have basically called a semi-permeable membrane. Well, that semi-permeable membrane is made up of something called lipids, which are essentially fats. So dish detergent basically dissolves fat. That's why it works. That's why when you put dish detergent in water, it foams and is able to wash, like you're able to clean dishes well, is because it actually dissolves fat. So what you're going to be doing is by adding the dish detergent, you're actually dissolving the membrane of your cells and the nucleus, because DNA is held in the nucleus of your cells, which is basically kind of like the brain of your cells. So that's where all of your genetic information is stored. That makes you, or makes fungus, or makes cats. It's all the same. It's basically what makes you, your own unique human being. So what you're going to do is you're going to just take this and you're going to invert it a couple times. You don't want to shake it to the point that you end up with bubbles, but you want to just kind of tip it up and down like basically what I'm doing right now. And what this is going to do is it's going to dissolve the cell membrane and the nucleus, like the membrane of the nucleus, and release the DNA from the cells. So in just kind of piggybacking off of this, while we wait for our little cells to dissolve here, is the reason why we look the way they do, why I have blue eyes, why other people have brown eyes, why you're right-handed or left-handed, is because of having dominant or recessive traits. And basically what that means is that some traits, it doesn't matter what genetic information you have, that genetic information will preferentially always show up in you. So for instance, brown eyes is dominant. So your genetic information will more preferentially select for having brown eyes than for having blue eyes. So in order for me to have blue eyes, for instance, both of my parents had to have blue eyes. If you have brown eyes, chances are you will have a parent that has brown eyes, because brown eyes are dominant. So that's just a little example of how everything, that makes us human beings is really just genetic chance. And those kind of things you can actually identify by just looking at your DNA. So now that hopefully we will have dissolved all of our little cell membranes, we can take this and take the cap off. And we're just going to let it sit for a couple more minutes while I explain a few more things about DNA. So essentially everything that you are physically, and everything that you can, even the traits that you don't necessarily express but are held in your DNA, are going to be able to identify from these things right here, which is why we can determine things like, why certain people carry certain diseases, or why certain people carry certain traits for things like eye color, or why when you watch shows like CSI or some of those other like Law and Order and whatnot, why they're able to actually, identify who committed a crime based on their genetic material, because everybody's genetic material is unique to them. So now the next thing that you do is you actually need to take rubbing alcohol. So DNA it dissolves in water, but it is insoluble in alcohol, which means that it'll actually turn into a solid when you introduce alcohol into the solution. So you're going to take your little pipette that I supplied you, and this I actually put in the freezer. So this experiment works better if you cool the alcohol before you use it, and you tip your test tube at a 45-degree angle, and then you will take your dropper and just lay it into the side here. So, and then you just very slowly release the alcohol so that it floats on top. So you don't want to be very aggressive with it and release it really, really fast. You want to be really, really gentle with it so that it floats on top. And then I'm gonna put some more in. So you take this and you, again, just very, very slowly put more on top. And you notice how the alcohol sits on top of the solution? And you notice that there's little bubbles forming inside? And you can kind of see that there's little wisps appearing. It almost looks like cobwebs. That is actually my DNA. So we're gonna put some more in. So this ends up being about, 2 to 3 milliliters of alcohol. And yeah, you can see that there's all these little kind of wisps appearing and that you get all these little bubbles. That's the process of the DNA actually becoming a solid in the presence of the alcohol. So you can see that there's some pretty obvious wisps appearing. So this is actually the scientific process of how we go about collecting DNA in a laboratory to do anything from criminal investigations to determining how, primates, like basically like humans and apes and all their ancestors are related to each other, to how, how human diseases are basically passed on from one generation to another. So yeah, this is a really neat experiment. If you want to keep it, you can. After a few more minutes, this will actually have a lot more genetic material that will appear in it. But yeah, try it at home and see how it works out for you. And if you want to, post pictures of your experiment in the comment section of this video, you can. And this time I won't see you next week because this is the last video, but it's been a pleasure, and I'm sure I'll be back at some time with more experiments.